a good article yet because I haven't started reading it. Looking at it, there are a couple of things that I want you to note when evaluating an article. First thing you're going to look at is what journal is it from, and I find that information right up here at the top. This is from the International Journal of Antimicrobial Agents. Now remember what my potential ideas were yesterday. I was thinking about how to use a plant extract as an antimicrobial. Do you think that that's a good journal to choose from? The International Journal of Antimicrobial Agents? Sounds very fancy. Very fancy indeed. So here's our journal. Looking lower on the page, right here is the title of the article again. Remembering anytime you have anything in italics where the first word is capitalized, the second word is not, biologically that refers to a what? Binomial nomenclature, genus and species. So we're looking at 1, 2, 3 different organisms here. This, and I do believe, if I'm looking at it, the 3 organisms that we are looking at, one is a protist, one is bacteria, and one is yeast. It's a fungus. So we're looking at 3 different kingdoms. Does that make it more interesting? Because we're not saying, oh, just bacteria. We're saying there are 3 kingdoms here that we can examine. So what do you think? For the purposes of my project, is it worth pursuing this article further? Okay. Then we look at the date of the article. Received the 30th of July, 2010. Is this new or old? Truthfully, it's a little bit old. We like to keep things within the 5-year window if we can. So this is just cutting it at the 5-year window. 5 years and newer, we consider recent science. Anything older than that is kind of old news. But 5 years and something that seems this relevant, we should probably keep reading. Another thing that we do is look at our authors. So here are our authors: W.L. Lowe, C. Martin, D.J. Hill, and M.A. Kenward. You'll notice there are no first names. Some publications, they remove the first name, so there is no bias about whether or not the researcher is male or female. There's no way of you knowing from those names. Bias is still very real in a lot of different disciplines, so this way we can say, we don't know whether they're male or female, doesn't matter to us. Some publications will still include the first names, a lot won't. If you look at them after their names, they have these little superscripts, right? A, B, C, D, A, B. That tells us what institutions they have done their work at. So here, our institutions are the University of Wolverhampton. I don't really know anything about the University of Wolverhampton. But it's in the UK, so that's probably why I don't know anything about it. But what departments are they from? Applied science, pharmacy, healthcare, biology, geography, environment in the School of Applied Science. Sound like people who might have a clue? Like their credentials are not like California Psychics. I heard a commercial for California Psychics on the way in this morning. It was very confusing. I was very confused. I just want to say it. No, you know what the problem is? When they were interviewing one of the psychics for the commercial to say, and her shtick was to say how rigorous and thorough the screening was for her to be hired, so it makes people trust you when they call in. So they're like, oh, it was very hard to get hired here. They really vetted the psychic. And the first thing she says was, I was surprised by how difficult it was to get hired. Well, how are you any good as a psychic if it surprised you that it was hard to get hired? Shouldn't you have known that?
I feel like it's kind of dumb to think of psychics, but if you ask just general questions in an area that applies to most people,
I will fully admit I'm a skeptic. I'm a scientist. I know you're shocked. You're like, what? She doesn't believe? Yeah, I know. But the whole commercial, I just started laughing. I was like, really? This doesn't make sense to me.
I don't know with scientists, I don't know with aliens, that's what shocks me.
But you would agree their credentials are better than the woman on the California Psychics commercial. No offense to the woman on the California Psychics commercial, but better credentials than that? Okay. We like to evaluate things like that because there are some articles that get published where people have pretty darn sketchy affiliations. Sometimes you'll read them and you'll realize that they're paid by a company. And then you're like, you were paid to do this research. I wonder if it was convenient for you to get the results that you got. Like, say, this was funded by a company that produces tea tree oil for commercial production. Then you would be like,
Are they any of like the students that help them with it?
Absolutely. Usually the first person that's listed is a lead researcher, and people who are listed after that are graduate student assistants, any students, anybody else. Sometimes they get credits in the credit line at the end. Absolutely. Keywords here: silver, tea tree oil, wounds, and our 3 different species. So they always kind of give us keywords. That's how we got this as a result for our search. Those seem like relevant ones? Okay. As we look at these things, we have to remember that what they're doing here is the scientific method. They're attempting the scientific method, and you know that scientific methods should have an independent and dependent variable. So it's not always as easy as reading it and immediately knowing. So as we look at the title and the abstract, I want you to consider what were the author's independent and dependent variables, and how did they measure them? 2 important questions. What are the variables, and how did they measure them? Guaranteed they were able to measure them because they produced a study and results. We just had to figure out how. And then we'll decide, is how they measured theirs, can I do that? So, you guys probably can't really see the abstract particularly well, right? That's good. It's like nice and cut off. Oh, I guess it's not that cut off. Let's see if I can't. Doink. Is that better? Larger? Slightly. Tea tree oil and silver ions, either alone or in combination with other antimicrobial compounds, have been used in the treatment of topical infections. Do we need to translate it? Or are you guys like, no, I'm actually good with that sentence? Excellent. However, there appears to be little data on the efficacy of TTO, which previously they told me is going to be tea tree oil. Efficacy of TTO combined with silver in the absence of any other agents. Still good? Any words there we don't know? Only one that's a little bit strange or new to us may be the word efficacy, but we talked about yesterday how that means like efficiency or effectiveness. Tea tree oil and silver, that's what they told us this is, right? Silver were added alone and in combination to suspension of cultures of Pseudomonas, Staphylococcus, and Candida. What was that sentence? They told me my independent variables, right? What were my independent variables? So we had a bunch of different experimental conditions, right? We did tea tree oil, we did silver, we did tea tree oil and silver. So those are our 3 variables. Do you think they did one without tea tree oil or silver? What do you call that? It's a control group. So they would have done 4 conditions here. They did tea tree oil, silver, tea tree oil and silver, and no additional antimicrobial at all. So 1 independent variable for 3 different tests, 1 control. Treatment of these cultures with tea tree oil and silver at subminimal lethal concentrations. Subminimal lethal concentrations. What's that mean?
Close to being, like not that close to being dead.
So just at that level where it doesn't kill them, but we want to see how it affects them, we're going to add at that concentration. So one of the things I'm going to point out about this is a lot of you are going to say, well, for my experiment, I want to add substance X to this. And I'm going to be like, you know what? That's not good enough. You need to determine a concentration. So instead, I'm going to suggest that you add different concentrations of substance X to whatever it is you want to do. That takes it from being to, yeah, that's suitably your age. It's going to require you to learn how to make certain concentrations, either molarity, parts per million, parts per billion. You have chemists available at your disposal to help you with those calculations and solutions. Turns out they're happy to help. Like Rumor, they get paid to actually help you. Yeah, so, yeah, it's crazy people. They show up every day and get paid to help you. Who does that for a living? Seems thankless. Yeah?
When you were saying, like, this thing when we first started, actually, you were saying how some of these things come up, like, really convenient answers for their, people are giving them money to write this and do all their
Researcher bias, right?
Yeah. And could they get in trouble for that if they're lying?
Oh, for sure.
If they say this tea tree oil can, like, cure.
Yeah, they have to print a retraction. And typically when you have to print a retraction, that kind of ends your career as a scientist. Like when you have to stand up in front of the scientific community and be like, ha ha ha, JK. They don't really let you publish anymore. Just essentially what you'd be doing.
Wasn't there the one guy and he was a scientist and he was working in the lab with like college students and he paid them to like,
And he made them fudge their results.
yeah.
Mr. DF's wife was instrumental in having him exposed as a fraud. Indeed. So that's bad news bears. He made his grad students who were recording his data, he made them record what he wanted the data to say rather than what the data says. We all say that and we're like, some of you will do the exact same thing for your science fair. You'll be like, well, Mrs. Carl and Mrs. Bloomfield are going to be so disappointed in this data. It doesn't say what it's supposed to say. It doesn't agree with my hypothesis, so I will just change this number here or there. Truthfully, we don't care if your data and conclusions match your hypothesis. We would rather you be honest. And if you get a no results experiment that you were honest, maintained your integrity and your scientific standards is better than one in which you fudged data and lied.
You get a bad grade if you have an.
No, you will get a bad grade if we find out that. I wonder what my husband wants. You will get a bad grade if we find out that you've lied, and we'll know you've lied because you have to take pictures the whole time. So. Really? The whole time. That's it's easy for you guys because you have like these things that have like cameras and stuff, right? Like, none of you can be like, oh, I don't have a camera. I can't do that. Yeah, but you can print from these. Take your pictures, save them to Google Drive, print from Google Drive on a computer. Easy peasy.
I think you still print off your camera roll. If you click arrow box out, arrow box.
Do you have access to one of those at school? Hey, there you go. Excuses abound.
I forgot to do my English paper, but I figured it out, so I just printed it out and
Who feels a teacher?
I strike you as a narc, don't I?
It's here. It's like someone who tells on someone else, kind of. It's like a snitch.
You're a teacher.
Yeah, and I'm like super scared of you guys because, I mean, snitches get stitches. Look how scared I am of all of you.
First
You're like,
hour by hour.
watch my back when I go out to the parking lot.
Before this year, I thought all teachers only talk to other teachers.
They're like teachers aren't allowed to talk to other people. Like they can only talk to teachers.
Yeah, I don't really have friends.
No, every time I see someone other than the teachers.
Um,
That's Mr. Burton, that's a sad one. He was like, I graded this for 6.5 hours.
That's
I
it.
didn't do
what does that thing I just underlined look like?
Kind of looks like how they did.
It's a result.
It's a result. So that's our dependent variable, right? Loss of viability. Loss of viability. Now the question is, we have to figure out exactly what loss of viability means, because I don't know how to measure that. I don't know what measurement you take for loss of viability. So we have to keep reading. Order of sensitivity to the combined agents was the Pseudomonas, then the Staphylococcus, then the Candida. So which one was more sensitive to the tea tree oil and silver combination? Order of sensitivity, where Pseudomonas is that direction, alligator is greater than or less than? Greater than Staphylococcus, which is greater than Candida. So Pseudomonas was the most sensitive, the one that was most likely to lose viability, which we still don't know what that means, but we'll get there. It's been like scary hard yet?
Yes.
I have a question.
George has got to say yes just because he can.
Yep.
It's because we didn't read Feast or Fast.
Yep. That's right.
And what's viability mean?
There's two definitions.
Let's hear them both and we'll decide.
Number one, the ability to live especially under certain conditions. Number two, capacity to operate what we sustain.
First one.
Well, they both sound kind of similar to me, right? Like loss of viability means you're not going to live anymore, and it means that you're not going to be able to reproduce anymore. Viable, to be viable means to be able to live. So loss of viability means you lose the ability to live. You lose the ability to sustain your system and any of your internal chemical reactions. Which, when you cause disease, that's probably like a good thing, right? We want loss of viability. The fractional lethal concentration index, fractional lethal combination index, one that sounds made up, right? Fractional lethal combination index doesn't sound made up to you. Sounds complicated, it sounds made up. It sounds complicated and made up because the researchers made it up for the purposes of their data analysis. They looked at the concentrations and determined where in those concentrations lethality occurred for each species. Or loss of viability, sorry, not lethality. That's it. Showed the combinations of tea tree oil and silver synergistic effect. What is a synergistic effect? Synergy, when things work together. So a synergistic effect is two things becoming more efficient, higher efficacy because they're working together rather than working independently. And an indifferent effect, so against the Pseudomonas, they worked together and it was better to use them. Against the Candida and the Staphylococcus, indifferent means, what's it mean if I say I'm indifferent? I'm indifferent, right? Like,
It would be which one
yeah, you're like, yeah, whatever. It's cool. I don't care. Doesn't bother me one way or another. That's what it means to be indifferent. And here they give us the rankings on their index, right? And they give us those in parentheses because it's important to present your data right away. The results indicate that combining these antimicrobial agents may be useful in decreasing the concentration of antimicrobial agents. May be useful.
What's the point of it to test it?
May be useful. What they're saying is, while our data showed that it worked for one of our 3 test species, it didn't make a difference for 2 of our test species. So it might work for some, but not for others. That's why they had to say maybe. You have made it all the way through their abstract.
Yeah, but there's still like so much more.
But you know what? I'm not asking you to read this so much more. You know what we do from here? Voila. We do what you guys do. It only, seriously, I only got the first page in here.
Oh, you said,
Ah. All we have to do, graphs, figures, charts. We look at title, authors, date, abstract, graphs, figures, charts. That's it. All those other words are only there in case of emergency. Yeah, those are emergency words. Like, if we're not sure how they measured something, we skim the emergency words for technique, and then we move on. Notice I didn't say we read every word of the emergency words. I was like, skim it for the bits we need, and move on. Yeah, that means that's not like a horrible task. You skim all the time. Quite frankly, this is exactly how you read when you're given a reading assignment for school. You read the title, you read the introductory paragraph, then you look at the pictures and you're like, yeah, I'm done. Am I right?
Yeah.
No, I attempt to read all of it. I actually get pretty far. I was proud of myself. 'Cause I
So-
push a lot of reading.
The pictures never give me.
I just read the caption.
Here's what we're going to do. We look at the first table, and it just tells us what's in the table. Microorganisms. The MLC is the minimum lethal concentration of tea tree oil in combination or alone. So the first one is percent volume by volume. That's a concentration. So that means that I put in like 0.1 milliliter versus 0.25 milliliters of the other substance, and I mixed them together. It's just a way to say how much of each you have. And then here, percent weight by volume. So the first thing we measured by weight, or in this case actually what they say weight, they mean mass. The first thing we measured by mass, so grams, versus volume, milliliters. You can measure grams and milliliters, can't you? Grams, you just need a balance, and milliliters, you need a graduated cylinder. You know people who are willing to loan you those things?
The bio teachers with Bloomfield's car.
Hey, just pay us a visit. We'll give you this little piece of paper, you sign it, and we send you home with stuff. Easy.
If you need it this summer.
We send it home with you all summer.
What does the
No,
paper
what
say?
if you, like, you didn't realize you needed it?
You'll know you needed it before, because by the time we're done with this, right before Memorial Day, you're going to have this whole giant list of every piece of equipment you needed. And then on the day of the final exam, you just stop by the SRC, come pick it up. I'll have it in a grocery bag with a Post-it note stapled to it with your name on it. And then you are on your way with all of your equipment. We make it easy peasy. I got a system.
It's like
I love
take out
systems.
down
Our closest Memorial Day.
The end of May. It's always the end of May.
Oh, March and
So we're, like I said, baby steps. I haven't even asked you guys to start reading yet. Baby steps. We're still reading my article. I'm not even going to ask you to read this week.
Whoa, the end of this was
Oh.
Are we gonna?
She's like, wait a minute.
Oh, she's
So-
right.
This then tells us the lethal concentration. So this is the minimal, the fractional, this is the minimal of silver. So here's of tea tree oil, here's of silver. This is going to be the fractional. This just tells us, all this table says, how much of each chemical they applied. Yeah. Okay, thank you. Thank you, researchers. It's just procedure put into a table. Done. And then they show us this fancy math problem for how they figured out that made up thing that I said they made up. It's just a math problem. They're like,
Is that called
let
an
me
algorithm?
math this for you.
Is that called
How
an
do we know it's
algorithm?
right? They made it up.
We don't.
So they could be fraud too?
Well, it's likely not fraud. It's likely just a way that you always have to find a way to do a statistical analysis. So this was their statistical analysis of how effective it was to be together versus alone. It's like you guys, for your statistical analysis, most of you are just going to do mean median mode. This is their version of mean median mode.
Can you do that? Do you have to go through like a board and say this is, like if they're gonna make it new?
They have to, yeah. You guys won't do anything that
And
complicated.
then they would have to, like the board has to accept it.
Yeah, so when they went to publish this, a whole bunch of really smart people read it, and they were like, your math is okay with us. Like, we'll accept this. So somebody else has read it and been like, that's a legitimate statistical
So like,
analysis.
do they like add that to the board? I mean, like the board,
Mhm.
list?
Is that a very well-paying job being on the board?
Not especially.
Oh.
These researchers make money usually by being professors. Even people who evaluate articles make money by being professors. So that's their first job, and this is what they do, because this is what they care about, and then they be a professor to, earn cash flow. Sometimes. Most of the ones honestly, truthfully, most of the profs I had in school who did this were exceptionally nice people, very down to earth, insisted that you refer to them by their first names, like very, very kind, amazing people. Occasionally you'd meet one who thought they were the bee's knees, but I mean, you meet people who think they're the bee's knees everywhere, so
What is the bees?
like awesome sauce.
Do bees have bees?
Yeah, that's what I don't get. Like, I know what it means, but
Where did it come from? I have no idea.
What does awesome sauce taste like?
Awesome.
What is the main defining taste of awesome? Like, is there
Glitter.
glitter?
See all these words? Words, 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 words.
That's how we should read all the time.
I'm my HSBC science version. I just
But instead, I'm like, who needs words when there's pictures?
Yes. But it's a very confusing picture. So
It's not such a bad picture. All we have to do is look at our pictures and figure out what they're saying here. So one of these lines is only tea tree oil. One of these lines is tea tree oil and silver. Another line is just silver. And the fourth line is our control group that doesn't have either. And then they're going to tell us time on one axis, so how long it takes. And this is going to be, CFU. Where do I find what that means? Colony forming units, right down here at the bottom in the caption. Colony forming units per milliliter. So what we do, to a microbiologist, this makes a lot of sense. They just take the plate that they cultured this on and they see how dense it is with colonies. The cloudier the material is, when you grow colonies, the more colonies there are. It's just a measurement that they use in microbiology that a microbiologist would know. So those of you who are interested in microbiology and you have a lab to work in, that's a measurement you learn. It's actually really easy. It's not a complicated one to take. You'll do it in undergrad if you go into science. I drew a line. That's okay, because you know why it's okay I drew a line there? We're not going to read those words! Nobody needs those words. So the first one is for Candida. Here's tea tree oil only. This is a good graph in that slope means something to us. Slope means rate of change. The change here, death. How long does it take Candida to die if it's only tea tree oil? Between 2.5 and 24 hours. Like after 2.5 hours, notice the break in the graph, they didn't include all of it. So it's pretty ineffective for the first 2.5 hours. Exposure has to be longer than that to be effective. Can you conclude that from this graph? Yeah, so it makes this an effective use of the line graph or XY scatter plot. How about for our representative control? No change? Is that good for a control group? Is that what you would expect? Yeah, if you're not manipulating a variable, it better stay the same. Here is, this is a circle, right? Or is that the square? I can't tell from here. Tea tree oil plus silver versus silver alone. So for Candida, what of the two antimicrobial agents do you suspect is the one that increases efficacy based on this graph? The silver, right? Because the tea tree oil alone, meh. Silver alone, a lot better than tea tree oil alone. But together, that's great. That's why they said synergistic effect in their abstract. Because they're like, hey, they work together, and they work together well.
Do we want it to go?
We want death to be down, right? Because these things cause disease.
Oh, yeah.
So death is good, which is,
So like for the
you're
control
like,
group,
what does that mean?
if you're doing like with like zoology, would you have like if you do 2 different variables and it's like something to do with like food, would you your control group you don't give them food?
You would give them, in those instances, you would have to choose a food type that would be the control, and then concentrations or level, however else you're going to alter it. You're not going to starve them, no. The two graphs below this, exactly the same, but this one for Staphylococcus and this one for Pseudomonas. Were those graphs so bad? Were they as bad as we thought they were when we looked at them? Do we know what the independent variable was?
Yes.
Do we know what the dependent variable was?
Yes.
Do we know how they conducted their experiment?
Yes.
Did we read all that long?
No.
Yeah.
Truthfully,
Out some of the words.
we didn't read all that very long. We look at the top of the next table, and it just tells us it's the summary of the correlation between the minimum inhibitory concentration and the minimum lethal concentration. So inhibitory means inhibits, makes it more difficult to do something. Lethal means kills you. They basically did a concentration study here. They were like, okay, at what concentration will it kill versus inhibit? So they did a little follow-up to their initial. That's all that is. And then, conclusion. I don't really need to know their conclusion because their conclusion is just going to be a whole bunch of words that go with the graphs. I care about the graphs. I don't care about the words. The graphs, that's enough for me. Acknowledgements, this is an interesting place to look just for the purposes of some ethical stuff. Competing interests, none declared. In other words, none of us who are authors are being paid by a company who produces tea tree oil. None of us are being paid by anyone who might make us biased. So they're saying this was just research in the name of research. I like researching. I'm paid to research. I have no bias. They have to declare that here. And they said none declared. And ethical approval, they didn't torture anybody. They just killed some microorganisms that don't have backbones or any way to feel things. They don't have to declare any ethical problems. Like, nobody was going to get sick. Nobody's going to get infected. If they follow proper procedure, life is good. That's all we expect you to do with Science Direct articles. We don't expect you to read all those words. All that stuff I scratched out, no expectation to do so. Abstract, graphs, figures, charts, title, authors, date, done. Not horribly unpleasant.
This is gonna sound kind of dark, but say that you're a question. allow you to test on a human if somehow the human was alive kind of back then? Like it sounds like weird, right?
The instances in which they allowed testing on humans who were injured, impaired, or otherwise unhealthy, one, the human has to submit themselves for the purposes of research. Some people do. Some people are like, hey, I'm a freak medical case. I understand that. Study me. Other times we did that would be like how we learned about digestion. Civil War, some guy got shot in the stomach. What does this doctor who's taking care of him do? Dropped food in through the hole of his stomach. We learned a lot about digestion in humans that way. But, you know, Civil War era, they weren't quite so worried about the ethical side of things. Other instances where we tested on humans, they were called Nazis. We don't do that anymore. It's true. None of you have heard like about the Nazi scientists, all that?
They were like two twins and they were like
Twin studies among the Nazis. There were in concentration camps scientists who would pull Jewish people from the camps and use them as lab rats, essentially. So when I say that, like I'm not joking. I'm not like, oh ha ha, they call them. No, I'm actually like serious. They were Nazis.
People think they tried to make Gorilla and hybrid warriors. The Nazis, people think that the Nazis, like they found some evidence that Nazis are trying to make Gorilla human hybrids to use in war.
Interesting. I got distracted by some of the words, but I don't expect you to get distracted by those words. I like that they then provide what they believe is an explanation for the behaviors that they noticed. You don't need to know that. Meh. Interesting? Yes. Do I care that you know it? No. This is the activity that I plan for you guys to be doing at the end of next week. You'll be doing it in class. You will have Thursday and Friday to do this in class with a total of 3 articles. That means by next Wednesday, you should have more than 3 articles. So if you do this and realize like, hey, this article is not what I thought, like you get to the abstract and you're like, this isn't useful, next. You can move on and not have to do the rest of this. You're looking for 3 good articles that are related that you can do this. Make sense? And then from there, we'll start posing research questions. What I'm going to do is next Friday, you're going to turn this in at the end of the hour. I have paperwork for you to fill out, so it's a method that you can follow, so you don't have to like make it all up on your own. You'll turn it in at the end of the hour Friday. I'm going to make photocopies of it, and then we're going to distribute that research to your cooperating teacher, whomever that's going to be, so we make that decision, next Friday. Distribute it to your cooperating teacher so they know what your research looks like, so when you go talk to them, you have your research in front of you, they have your research in front of you, and you can have a real scientific, educated conversation instead of just a, hey, do you think I could? Because they're not going to entertain that with much patience because they've got like a bajillion of you that are going to walk up to them and ask them questions. This is where I also tell you, now that we're starting to establish our cooperating teacher relationships, please, 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 my friends, when you go talk to them, ask them if they have a moment to talk. Say please. Say thank you. Ask them if you can schedule an appointment to talk. The reason you're not technically their student and you are going and asking them for their spare time. Have they already agreed to do it? Absolutely. But, it's polite. And for the record, I'm usually told at the end of this process that my students are always the most polite. And that makes me happy. So, be pleasant, smile. Don't go to them like it's a burden. Be like, hello, stranger. Probably me. So.
If you're not polite, this week, who's gonna hunt you down?
It's probably not.